As we continue our journey through omics strategies, we have thus far covered metabolomics, where we look at small molecules typically with molecular weights of less than 1,500 Daltons, and we focused on proteomics, where we are looking at the proteins present within a cell or within an organism. Now, to continue our journey through omics, we are going to be looking at genomics. And in order to discuss genomics, the first thing we need to look at is genome sequencing, determining the exact sequence of DNA that is present within an organism. And so, as we look at this schematic, you may be wondering what in the world we are looking at here. What we are looking at here is what we could think of as a chromatogram of the different nucleotides that make up DNA. So as a result of a genome sequencing experiment, one would generate this sort of trace where the different colors of peaks in this representative trace correspond to the different nucleotides as listed above, enabling us to put together a portion of the genome of an organism. So when we are looking at this, we're interested in sequencing the entire genome of an organism or a portion of the genome of the organism in cases where perhaps we hypothesize that a particular gene mutation resulted in a disease. We might be interested in just sequencing that particular gene rather than sequencing the entire genome. When we consider the sequencing of an entire genome, the genome size differs wildly between different organisms. For example, the genome of E. coli, a bacterium that's very common in your gut and also very common in a variety of laboratories as a model organism, has a genome that is approximately 5 million base pairs in size. If we think about instead, say, a simple fruit fly, the size of the genome jumps up to about 123 million base pairs in size, and a human genome is 3,000 million bases, or in other words, 3 billion bases in size. So there are definitely some extremes to the spectrum of genome sequence size. Interestingly, as a fun fact, there's also some surprising extremes. We would think of humans with a 3 billion base genome as being likely the most complex genome on the planet, because we consider generally ourselves to be very complex organisms. However, there are genomes that are larger than the human genome, such as that of the loblolly pine tree, who has a genome that is 23 billion base pairs in size. In other words, that pine tree has a genome that is more than 7 times larger than the human genome. When we are thinking about sequencing these genomes, the cost and difficulty associated with sequencing a genome is very dependent upon the size of the genome. The smaller the genome, generally, the cheaper it is to sequence, and the easier it is to generate a complete genome sequence where we know every single nucleotide identity within that particular organism. Considering the human genome as an example, because understanding of the human genome has been incredibly informative in terms of diagnosing and treating diseases and understanding diseases. When we think of the human genome and its 3 billion base pairs in size, the first human genome sequencing project was declared complete in 2003. So about 17 years ago was when the first human genome project was declared to be complete. Now, keep in mind that sequencing project was for one representative human, and although it was called complete, in actuality, it was reported to cover just about 99% of the entirety of the genome sequence, so very, very close to complete. But keep in mind that sequencing the genome of just one individual human is not adequate for things like prediction of disease or understanding why there is such variability of traits across humans. And so in order to understand that, what was necessary was comparison of genomes across different humans. And in the case of the first human genome sequence, sequencing was extremely expensive. We look at a plot of the cost of genome sequencing. What we can see with this cost of human genome sequencing is that they look over here at the left-hand side of the plot, starting around in 2001 to 2003, when the first human genome was considered complete, the cost of sequencing a human genome was approximately $100 million. Super expensive, cost prohibitive to do more than just one or a handful of human genomes. Fortunately for the progress of science, the cost of sequencing the human genome has dropped dramatically, following this green line as we go through, moving onwards to 2020, where the cost of sequencing a complete human genome has now dropped below $1,000 per genome. So with this dramatic drop in cost of genome sequencing, the use of genomics to address questions related to disease and related to biology, including human biology, has dramatically increased with this decrease in cost. So we can ask the question of why has the cost decreased so dramatically over the course of the last less than 20 years? The reason is that if we roll back 20 years ago, the main state-of-the-art technology for sequencing genomes was what's referred to as Sanger sequencing, which you may be familiar with from some of your previous biochemistry courses. This is the traditional main method of DNA sequencing. It is relatively slow, low throughput, in other words, because of the fact that it is slow. It's not suitable for sequencing a large number of genes or genomes really quickly, but it is generally quite accurate in terms of the individual nucleotides that are predicted from that sequencing. Over the course of the last 20 years, the reason why the cost of genome sequencing has dropped so dramatically is because a variety of technologies have been introduced for what's referred to as next-generation sequencing. Next-generation sequencing is generally referred to as NGS, for next-generation sequencing. And a huge variety of different technologies have been released for next-generation sequencing that have enabled a lot of competition in the market, a lot of increase in the throughput and speed with which sequencing occurs, and resulted in a drop in the price of each individual genome sequence. So next-generation sequencing has very much revolutionized our study of genomics because it has enabled the sequencing of individual human genomes at a relatively low cost. And with that dramatic drop in cost, that has enabled the sequencing of a sufficient number of genomes that we can now compare genes present in a variety of different individual humans to correlate the presence of certain diseases with certain genes, or specifically certain gene mutations where individual nucleotides have changed from an organism that is healthy to one with a disease, thereby increasing dramatically our understanding of biology. Now, when we consider next-generation sequencing, which is going to be our main focus here when we think about sequencing techniques, because it is the next-generation sequencing that has very much enabled our study of genomics. In next-generation sequencing, we can break the field of next-generation sequencing into two different types of overall sequencing. One referred to as short-read sequencing, which focuses on next-generation sequencing techniques that provide nucleotide sequences that are between about 35 and 700 nucleotides in length. These short-read sequencing experiments rely upon generating a huge number of these short reads from different portions of the genome to ultimately assemble those tiny reads, those tiny short sequence fragments, into the entire genome of an organism through this so-called short-read sequencing technique. And as you can imagine, if the max length of a particular fragment of the genome is 700 nucleotides, and we're thinking about an organism even as simple as E. coli that has a genome that's 5 million nucleotides, it requires a huge number of those short reads to be linked together to form the complete genome of a 5 million nucleotide organism. Long-read sequencing will produce nucleotide fragments that are longer than 700 nucleotides, generally not the entire length of the genome of an organism, but instead fragments of the genome that are longer than 700 nucleotides that can then be assembled by linking the different fragments together to form the complete genome of an organism. Now in 2020, with next generation sequencing having been developed over the last 15 years or so, there are a variety of so-called short read sequencing techniques and long read sequencing techniques as well. We aren't going to go into each of those individual techniques, but suffice it to say that there are a variety of different companies in these markets that have created patented proprietary sequencing methods that give these so-called short reads or long reads. In each case, once a sequence is generated using the short read or longer read sequencing, some additional steps are necessary in order to utilize these data to address questions related to the genomes of organisms. So let's take a look at what one would do once they've generated next generation sequencing data using either the short read techniques or the longer read techniques. So next generation sequencing data analyses is what we're going to take a look at here. And the overall process here is analogous regardless of whether you're dealing with a short read or a long read. The specific softwares that are used are dependent upon exactly what technique was utilized to generate the data, be it a short read technique or long read technique, and even within each of those individual categories, the specific software that one would use for these upcoming steps is dependent upon what manufacturer generated the equipment for the next generation sequencing experiment that was used. And so as a result, I'm not going to talk about specifics of individual techniques for next generation sequencing data analyses, but instead focus on a broad overview of the general process of how this works. So once next generation sequencing generates some fragments of the genome, generally this is not going to be a single fragment that encompasses the whole genome, but instead what will happen is that there will be multiple so-called reads of various parts of the genome. So the multiple reads correspond to fragments of the genome from a variety of different regions of the genome, assuming that this is being done in an untargeted way. And so if we were looking at short reads here, these reads would correspond to somewhere between 35 and 700 nucleotides. If we're looking at those longer read NGS methods, they would correspond to nucleotide sequences of 700 nucleotides or more. Once we have those reads, those by themselves are not generally particularly useful, but instead they need to be assembled. So what happens then is, assisted with software, the individual reads are assembled into what's referred to as Contigs. The Contig is what is formed by linking together multiple reads into a longer continuous DNA sequence. So, for example, as a simplified representation of this, hypothetically, if we had read 1 and the nucleotide sequence that was determined for that read number 1 was this. I'm just writing out a thread of nucleotides here. And then hypothetically, we had read number 2, which had the sequence that I'm going to show here. What we would see by comparing these two is that there is a region of overlap. Namely, if we look right here, this particular sequence that I've highlighted in green for read number 1 matches up with what we see in read number 2. And so as a result, those two fragments of the genome would be assembled using specialized computer programs that are designed for whatever next generation sequencing method you use to generate these data, to create an assembled contig. The contig is what results, again, from joining together multiple reads. So linking those two together would give us a more comprehensive view of the genome. And I'm going to change to green right here because we're getting into that region that overlaps between the two sequences that allowed us to link those two together. And then continuing onward, just copying down exactly the sequence that was right here toward the end. We generate this contig. And then ideally, some other read would link on to this end of our contig and to this end of our contig as well to extend that out even further until ideally, in the perfect scenario, ultimately we would assemble a contig that corresponded to the entire chromosome of the organism. In the case of the human genome, that would correspond to one of our 23 chromosomes. In the case of bacteria, bacteria generally only have one chromosome, often that's a circular chromosome. And so if we were able to assemble a contig that represented the entire genome, that would be one continuous contig from that bacteria. Ultimately, in real-world genome sequencing and analysis, oftentimes what is generated rather than a complete genome that contains the entire genome sequence of a particular chromosome is what's referred to instead as a draft genome, where some of the parts and pieces are missing. So after the assembly of the contigs, once we put together all of the contigs that we can into a scaffold that contains all of the DNA sequencing information that we've generated from these next generation sequencing experiments, what is generated from that is the genome. And the genome can be either the complete genome, where the complete genome has one contig in total after we've assembled everything, representing the entire sequence of the chromosomes. Or alternatively, in contrast to the complete genome, what may also result from this assembly of contigs is what's referred to as a draft genome. Where much like in the rough draft of a paper, there are some parts or pieces or minor points missing. In the draft genome, ultimately what we generate is not just one contig that contains the entire chromosome sequence, but instead there's some contigs that can't be linked together because there's no overlapping nucleotide region, and so multiple contigs are present, representing some portion of the entire genome. And depending upon the exact amount of coverage of that total genome, this could represent nearly all of the genome, or it could represent as little as half of the genome, where the other half of the genome had been completely missed. And in the cases of the creation of a draft genome, if one were to desire to go from the draft genome to the complete genome, what would be required is just additional next generation sequencing. And often, from a practical standpoint, what's necessary to go from the draft genome to a complete genome is additional next generation sequencing using some next generation sequencing technique that is complementary to the one used initially. So very commonly, people will do a hybrid sequencing experiment where they sequence a genome using short read sequencing techniques to generate a bunch of fragments of 35 to 700 nucleotides, and they'll complement that by doing a long read experiment where they generate fragments of sequences that are greater than 700 nucleotides and assemble all that together to ultimately create a complete genome sequence for an organism. Now, once we have either a complete genome sequence for an organism or a draft genome sequence of an organism, what do we do with that information? That is going to be the topic of our next video. Is now that we have got to the point where we've generated genome sequences, what can we do with that information? How can we analyze those data and utilize those data for some specific example applications?